Hey, everybody, I'm here today with a really special guest. We have Dr. Amy Proal, who is a microbiologist and a very, very special person that came recommended to us by a lot of people. And we're happy to finally have her on. So, Dr. Proal, welcome to the Tick Bootcamp podcast.
Thank you, Matt. I'm glad to be here.
So, Dr. Proal, can you tell us, start by identifying what you do? And I want you to be a little bit of a show off here because you're a really cool person. I don't want you to be humble. Tell us what you do and how you're helping the tick-borne and Lyme disease community.
Okay, well, I have two main jobs. So in 2018, I co-founded a nonprofit research organization called PolyBio Research Foundation with actually my life partner and neuroscientist. His name is Mike Van Elzeker. What really started us starting this foundation was we were both at a chronic fatigue syndrome conference that year in 2018. And I'll just be honest with you, the talks there were not great, and there was nothing on root cause. And we were both really interested in infectious contributions to why people could develop chronic fatigue-like symptoms, and this was just not at this conference. And so I have a microbiology background and I don't know, a huge amount of experience and interest in what bacteria, viruses, parasites do, the mechanisms by which they can drive a wide range of different chronic disease processes. And he has, in my mind, an encyclopedic knowledge of the brain circuitry, the way the central nervous system functions, and actually a lot of knowledge on infection. And so we merged this together to kind of figure out if we could create research programs that we thought would get at the root cause of chronic fatigue syndrome, and other conditions like that that we think have infection, honestly, at their root. And so we built PolyBio to do that. And what we do is we're not a typical foundation in the sense that we don't put out like a call for proposals. What we do is we're scientists ourselves, and we just network with people in our communities and essentially convince them to study chronic Lyme disease or chronic fatigue syndrome or babesia or long COVID, which we've done a huge amount since the pandemic. And what we do to bring talent, like new talent into these spaces is honestly network with teams that have done a lot of work on HIV, for example, and the diagnostics that really helped identify, HIV when it can be deep in a person's tissue and not always identifiable in their blood, or with other, pathogens like Mycobacterium tuberculosis. And we pull from those communities and bring those people into this space that sometimes people call infection-associated chronic disease. And we built PolyBio to make that happen. And so I lead that foundation in our research programs, which include a whole consortium of really cool global researchers studying the long COVID disease process, including one of the top drivers of long COVID, which is the persistence of SARS-CoV-2 in a person's tissue, where people just don't clear the COVID virus sometimes from the tissue of their gut or other body sites. And increasingly pivoting a lot of those teams that study, pathogen persistence or tissue-associated infectious processes into chronic Lyme, into the other co-infections in that space as well. And that's my first job. And then my second job is I'm the scientific director of the Cohen Center for Recovery from Complex Chronic Illness, or the CORE. And it's a clinic at Mount Sinai that was founded about 3 years ago with my amazing colleague, David Petrino, who's a innovation expert and honestly is very good at just knowing how to cut through bureaucracy to make things happen. And it's a clinic where we're innovating care for patients with long COVID, chronic tick-borne, vector-borne illness, and we're running an increasing number of clinical trials at that site as well. So those are my
Wow.
things that I do
Can
in the
you,
space.
Dr. Pearl, can you repeat that? So you said you're the scientific director of the Cohen Foundation Clinic, but I missed some words in there. Can you repeat the name?
Yeah, it's the Cohen Center for Recovery from Complex Chronic Illness, and we call it the CORE for short.
Got it. And is that related to the Cohen Foundation? Is that why it's named
Yeah,
Cohen? Okay.
it is. We're very thankful they support it, along with several other really great private donors. But it is, first and foremost supported by Cohen Foundation.
Got it. Okay. So what I really like about your PolyBio Research Foundation is it's a not-for-profit, but it's run by two brilliant scientists. And sometimes you see these not-for-profits be formed. They are run by a board of executives who are just business focused and don't understand the patient experience and really what the need is out there. And they're raising money, but they're spending money in ways that may not be appropriate because they don't fully understand the disease and what it's like to be a patient and what's needed. So I always love to hear when these not-for-profits are being run by people who are researchers themselves and scientists themselves like yourself, but also people who've been impacted by chronic illness like yourself, because you need that experience to be able to make informed decisions about how to run your not-for-profit to effectuate the best change possible in the world, right? So that's really exciting for me to hear that piece of it. And
Yeah.
then as a scientific director of the Cohen Center for Recovery from Chronic Illness, what is your primary role there? Are you actually seeing patients in a clinic? And, can you give us a little bit more about what you're doing in that capacity?
Yes. So what our first goal is we are seeing patients and we're creating as much as we can a precedent to be able to see patients with long COVID and chronic tick-borne vector-borne illness in an insurance-based system, which I'm sure you know is a challenge,
Oh,
right?
yeah.
Because we know there's a lot of expertise, especially in the chronic tick-borne vector-borne illness space. There's a lot of Lyme literate physicians, a lot of knowledge of what can be done. But as all of us know, a lot of that knowledge sits in practices where it's understandable. People have to cash pay because it's very hard to spend a lot of time with the patient, which is often needed. That being said, we are we really want to be able to create at least a basic standard of care for patients with long COVID and chronic tick-borne vector-borne illness in the system. And so that's where we do a lot of work, either running clinical trials to set precedent for what therapeutics can work most in these infection-associated chronic conditions. Because if we're able to run a successful trial or a successful research study, that allows us to fight more for insurance coverage of the treatment or of something relevant to patient care. So that's one of the things we do is we try our best to trial the things that we think could become used more in mainstream and fight for insurance coverage. And then we also work on, billing codes, things like that. So we're trying to innovate the mainstream, if you will, which is a challenge. And that's what we're doing. And we are treating a large number of patients now with long COVID and gradually more with chronic tick-borne vector-borne illness as well. And like my role is David Petrino and the medical director, Joseph Herrera, work day to day with the patients. And David works some on the research, but I come in and we take advantage of the fact that when patients come in, for a regular visit to see the doctor, that we can say, hey, as long as you're here, would you like to participate in a research study or would you like to participate in a clinical trial? And we can then do further work to help understand even more about what works in terms of root cause drivers of symptoms or treatments. And so as a scientific director, I just sit on top of patients coming in and I'm like, what should we do? Should we get, a skin punch biopsy and look, see if we can better understand Bartonella persistence in the nerve of the skin? Or, I kind of conceptualize the research that we can do as basically a kind of joint hybrid research clinical center, which is what we are.
That is fascinating. So we're going to come back to this, but I want to talk more about how you landed in these two awesome roles. But I know people listening are going to be dying to know, is this something that is open for new patients, the Cohen Center for Recovery from Complex Chronic Illness? Because many people listening are desperate for help and they want to know if this is something they can potentially look into from themselves, especially if it's covered by insurance. So is this something that people could reach out to their website? And we'll put the link in the show notes if so, to potentially come on board to treat with you guys and also potentially be a part of a research study?
Yes, definitely. We have research studies and trials that are enrolling now, and I can send you some of those links. We have somewhat of a waiting list for patient care. It's not terrible, but there is a waiting list. And to try to deal with that, we are creating a medical education. I mean, our best hope is, as you know, we'll never be able to see everyone in our one clinic. So what we're trying to do as well is create a medical education program where we start to teach physicians at other sites what we're doing. And our first pass at that, and we have many more plans to extend this, is a manual we created, which is actually on the Lyme HHS website right now. It's an infection-associated chronic disease provider manual. And at least we wrote down how our clinic operates, what we do when we bring people in, the testing and the treatments that we, prioritize. But we're building that out as well. So in the meantime, though, we are taking new patients and we can put you on a list if you're in the New York area.
Wow. So this infection-associated chronic illness provider manual, is this a manual for patients to find other providers, or is it a manual for existing providers to learn to be more Lyme aware?
It's existing providers to learn. And right now, the majority of the manual is long COVID because we actually started the way that I began working with David Petrino at this center was because he actually runs another clinic at Mount Sinai focused on rehabilitation innovation. So if someone has a stroke or other events, they innovate the tech and the care for those patients. It's a really well-known center, the Abilities Research Center. And when the pandemic began, COVID, patients who didn't recover from COVID just started coming into that center and saying, I'm still feeling sick after COVID, really, really, really sick. What do I do? And so David eventually just didn't want to turn those patients away. So he was like, I'll just start a long COVID clinic too. And he will admit that he really didn't know much about long COVID and just rapidly started to learn and to network with other people in our Polybio consortium on the research, which he joined. And so we built the clinic out first in almost just an emergency state for long COVID. And then we started pivoting into chronic tick-borne, vector-borne illness, which is a little harder even because of the difficulty within mainstream of having to push back against guidelines and having to sort of re-evaluate some of the guidelines that are set that we might need to expand on.
Yeah, for
So it's
sure.
a little bit, it's a work in progress. But yeah.
Now, so obviously those probably everybody listening has heard of, at least if not watched, the Lyme working group that the HHS department at the US government did. And they created this new website, hhs.gov/Lyme. And I think you said that your guide, the infection associated chronic illness provider manual, was actually linked to on their hhs.gov website. Is that true?
It is, yeah.
That is so good to hear, because Rich and I always say that there's so many great people in this space, but they need to work together better. So to hear that PolyBio has partnered with the Health and Human Services Department of the federal government to advance the cause is really empowering to hear, because we need people to work together, and together we can have greater influence to, effectuate change to get people better care that they need. So that's awesome. Now, I do want to ask, this is my final question before we circle back to this. I want to get into your background a little bit. If somebody's listening and they aren't able to travel to, what is it? Is it New York? Is that where the facility is, in New York City? So they can't come to New York City. Do you guys do any kind of virtual sessions for, in the clinical environment for patient care?
Not yet.
Okay.
No, unfortunately.
And that's people, I think many people will travel. And if you hopefully will consider doing some virtual care down the road, because I think that'd be a great service to people. So let's back up to, how the heck did you get here, right? I mean, obviously you're a brilliant microbiologist. You are an expert in the field, but what led young Dr. Amy Proal into this space? Give us a little context of what your life was like as a kid and how you ended up here.
Yeah, I mean, I had a great life as a kid. I grew up in Mexico City, Mexico. My dad's family was French, but had emigrated to Mexico City years before, started a tool manufacturing company that was cool. And my mom was just a Spanish student that came down to Mexico, met him, and stayed there. So I grew up, I went to an international school there. I had a fraternal twin sister, was my best friend, and it was very good. That being said, between the ages of 3 and 5, I got a series of really terrible infections. And one, despite being vaccinated for MMR, one was deemed measles. And I did go to a very good hospital for this. One time I was rubella. I was hospitalized again for that. I was hospitalized for a bad case of pneumonia, a bad case of scarlet fever, and then for one case of meningitis that was never determined what it was. Okay. And then I went on to be the captain of my tennis team and beat boys at sports. Really, I didn't have many issues until maybe the end of high school and towards the end of college. I don't know. I got headaches. I was like, I don't know. I totally just tried to not focus on the fact that I maybe didn't feel as good as I should. And I was a pre-med student in college. And so I was, doing really well in those classes. In fact, leading my class and learning about medicine and planning to be a doctor. And then right near the end of completing college, I got another infection. And I really got sick. I did not do well. I got very, very sick. And so, as I'm sure a lot of people have had, I went to the student healthcare center and I was like, hey, I don't feel well. And they're like, hey, we ran your blood work. It's fine. I was like, are you sure? Yeah, and it went quickly down a path of they referred me to a psychiatrist, and the psychiatrist started telling me that it was in my head, the typical path, right? But very quickly, And I think it was because I had such a great childhood and was pretty confident that I was doing okay. I was like, this is ridiculous. Like, of course I don't have some mental issue. And of course that infection that I got matters, even
Yeah.
though people would tell me that it didn't. And what I found fascinating from the beginning was that I still felt like I had an infection. I mean, I had, my throat would feel really sore during periods and my symptoms would go up and down. I'd feel kind of feverish. And so I was like, could I still have the infection in me? Maybe it's still in my body. And I would tell this to people, and of course they'd be like, no, we ran blood work and, we don't have it. And
Hmm.
I would think then, well, couldn't it be in my tissue? Couldn't it be in my brain? How do you know from your blood test that I can't have something in my brain? And that just led me down a path of, at that point I had to stop everything. But from my bed, I would read a huge amount of scientific literature. And I became really interested in papers that were written in the 1960s, the 70s, before especially the theory of autoimmunity gained hold in a major way, which was not a great time in science. Before that, there was a lot more exploration of what microbes could do to drive a range of different chronic disease processes, everything from, schizophrenia, neurological symptoms to Alzheimer's disease, all kinds of different chronic conditions. And these really great papers that would sometimes have images of persistent bacterial organisms and parasites in them where they were culturing them under different conditions. They were really interesting in terms of even mechanism. There was a paper by a guy named Gerald Deming, who was a wonderful researcher at that time, and it's called Bacterial Persistence and Expression in Chronic Disease. And what they would do is they would actually take certain types of antibiotics and show that when they treated bacteria with these antibiotics, the beta-lactam antibiotics, many of them would die, but some would lose their cell wall, become a different form, almost a different shape, and survive in the face of the antibiotic and persist, right? And I was like, this makes a lot of sense to me. It doesn't make sense that a single round of antibiotics or any of these things would kill off these incredibly crafty and often very persistent organisms. And so I already started at that point what I still do now, which is I contacted Gerald Domingue. I asked him if he could, I think it was at the time Skype me, befriended him, got him to tell me more about his work, got him to teach me. And then gradually I began a protocol of just low dose pulse antibiotics, not even knowing what I was treating, but
And
just
this is
thinking that it
I'm
made
sorry,
sense.
Dr. Proal, to
Yeah,
interrupt real quick, but
yeah,
this is now you're at the end of your college
yeah,
career, about to
yeah,
graduate to be a
yeah,
researcher, and you're basically self-diagnosing
yeah,
yourself by collaborating with peers before you even have your MD
yeah,
and your doctorate, and
yeah.
and all while sick.
Yes, I was sick, exactly. And I was trying to just, learn my way out of it.
Yeah,
Um, yeah.
and I just want to say that like all of this is so it's a little frustrating to hear because I can't help but think of we just did a social media post about the late Dr. Alan McDonald, who we had on the
Mhm.
podcast twice.
Yes.
And we have on both of our podcast interviews, he's provided PowerPoint slides that he's prepared for the Tick Boot Camp podcast.
Hmm.
And he puts images from his, microscope that he literally real images of the Lyme bacteria in
Exactly.
brain tissue, in heart tissue, in tissue, not just in blood. And he had studies showing that the Lyme bacteria persist in various organs and tissues for over a decade with even consistent antibiotic treatment. So this
Mhm.
evidence has been out there since the 80s, but
Exactly.
it's been refuted. And he went to various conferences, presented his data. He's been accused of lying and fabricating his information, and it wouldn't be well received. So, it sounds like you were coming on the same findings, but it just wasn't well
Mhm.
received by the community. But yet you were struggling with a similar illness and you didn't know what the root was from a pathogen standpoint, but you knew there was some sort of underlying chronic infection driving your symptoms.
Yeah. It got to the point where I even have a bookshelf of books of different scientists like Alan McDonald who were studying. For example, there's a book called The Cancer Microbe, a wonderful researcher who spent her whole life looking at the role of bacteria in cancer and again, just couldn't get through, left me all her books, was like, you're the keeper of the keys to my kingdom, just a whole career of knowledge on this that was not, didn't really hit mainstream, right? So there's a huge, huge amount of work that was done on persistent infection and chronic disease that is
Yeah.
out there. But,
And
and yeah,
and it's a misnomer too, as well, to
yeah,
say that the infection has to be in your blood. I mean, there was a study
Oh
last
yeah.
year that came out, and we've discussed this with various other doctors and researchers, and I think I know where you're going to land on it. But some doctors will push back and tell me that this is flawed. But the research study basically said, hey, there was this patient for a year was testing for Lyme disease, never tested positive with standard serology, right, blood
Mhm.
testing. But then they did a CFS test, and they found the Lyme bacteria because it was more in the neurological system than it was in the blood. And they said, oh, that's extremely rare, and that must have been an anomaly. That never happens. If you have a Lyme infection, it's going to show up in the blood, but it doesn't always show up in the blood, right? And that's the key
Mhm.
here. And the testing is really poor to begin with. So
Yeah.
even if it was in your blood, maybe the testing isn't going to find it. But even if it was, it's that whole thing of it's not rock solid, it's not definitive. And I think that's part of the struggle that
Right.
we run into with situations like yours.
It's a huge thing. In fact, generally speaking, if you're a pathogen and you infect someone and then you're going to persist in their body, the blood is not a smart place to be. That's where a lot of the immune cells are. It's where you're most going to be recognized. It's where you're most going to be targeted. So a lot of pathogens over time will go hide in a person's tissue and just dig a little bit deeper into tissue or nerve where they're, actually somewhat more evade immune recognition. And we even call that with some pathogens, when we're studying now, even SARS-CoV-2 can do that in some patients with chronic symptoms. We call it, in that case, the SARS-CoV-2 reservoir, where a little bit of the pathogen, again, basically stays hidden in your tissue, despite the fact that it's not widely circulating in your blood where it can be measured by a standard test. And there are, and that's a big thing that PolyBio Foundation is focusing on innovating now. There are diagnostic test platforms that might be able to capture tiny amounts of the pathogen or the proteins it makes in your blood, but those aren't out there now. They need to be built and iterated. There's a lot of labs, for example, that can find single, let's say it's SARS-CoV-2, single viral particles in an exosome, which is a little vesicle that might leak out of tissue, right? And capture that. But you need new technology to do that. The current tests that doctors run stand no chance. So that's the thing that is very hard. Yeah, with
Is
these
that similar
cases.
to like with Lyme disease, where they say a lot of research studies say that it's especially recently it's pooling in the liver. So after an infection or when it becomes chronic, it's seen in the liver a lot, but not in the blood. I think that's a similar example. It just kind of it'll go into a particular part of your body, and then you'll never test positive, but it's still
Right.
causing chronic symptoms, right?
It's very possible. Even in acute Lyme, Borrelia, it's actually even accepted in mainstream medicine that direct diagnostic testing to look for just the genetic material of Borrelia in blood is not reliable. It's that much of a tissue associated organism that even in acute infection, people don't think they can find it in blood, let alone in chronic disease where it's had time to, disperse throughout tissue, right? So yeah,
So
that's
let's
part.
circle back to your experience. So you're in college, you're now collaborating with other scientists and doctors to figure out what's going on with your health. Tell us what happens do you graduate and how do you continue to progress with your own health experience?
I did. I took low pulse antibiotics and did a couple other things and started to improve somewhat enough. And I got my degree, not MD though. I got my undergraduate biology degree. But then I decided I didn't want to be a doctor because I didn't think doctors had that much to run on, and that I thought it would be better if I became a microbiologist so that I could understand and create more evidence for what might be actually happening with patients with infection-related chronic disease so that doctors would have more to go on. So I changed to be a PhD microbiology path. And as part of doing that, what was huge for me is this was around 2005, this is a while ago, where, you know, like a decade before, the human genome had been sequenced with new computer-based tools that were able to take human genes and use computer tech to identify the genes, which was amazing to me. And the teams that had done that were starting to innovate that to be able to find microbes in the human body also with computer-based tools. And what they can do is, like, you can pull out of a tissue sample all the genetic material, and you can use a computer to identify what isn't human. And based on the sequence of the virus or the bacteria, you can call it and name it and know it's there. So this was happening at the J. Craig Venter Institute, which is a research institute in San Diego that we still work with. And I went to one of their first ever sequencing courses on finding new bacteria in the human body based on sequencing methods. And what blew my mind is there, in our first class, we identified several new bacteria in human samples that had never even been named before. And this was at the time where the human microbiome, even the bacteria in the gut and all of these organisms had barely been characterized. And so what it also created in my mind was this huge open-mindedness to say, look, there's these pathogens that we've already characterized, but a lot of our knowledge on that has been teams that have had to grow them in kind of like a classic, like lab coat petri dish setting. And so many of the organisms that can even make us sick or interact with our health and disease states in our bodies don't even grow under those conditions. And it became this revolution of using new methods to even identify other organisms, bacteria. So what it really did was create an understanding to me and the people I worked with that we were not closed-minded to the role of organisms in any kind of disease process. If anything, it's just a place of discovery.
Right. So basically, we don't know what we don't know. And
Exactly.
in this discovery in 2005, when we're starting to scrape away, I like to think of it as just scraping away the human component and leaving just the
Exactly.
non-human component, you're finding
Yes.
bacterial samples that we've never even knew existed before. And
Yeah,
that's a continuing process that we still see to this
exactly.
day, over 20 years later from that 2005
Exactly.
discovery.
Right.
And look, we're seeing it with Lyme. We keep track of how many species of Lyme disease are there, or
Exactly.
how many, depending on what you call it, subvariants. And
Hmm.
the number keeps changing because,
Exactly.
there's so many different types, and
Mhm.
each different type of the bacteria can cause different symptoms. Some are
Exactly.
more neurological, some are more rheumatological, right? So that's the difficult part about this. But in time, we'll learn more. But I think you're right. The approach has to be, we have to be open to the fact that microbes can be causing illnesses that have an unknown origin. And I think that fibromyalgia, ME/CFS, all those things have unknown origins. And I think a lot of those things are related to pathogens, which is exactly what you're focusing on. And what's really cool to hear is you basically redirected your career based on your own health experiences to become a researcher to get
Mhm.
deeper to the root instead of being an MD, because you felt you can help more people by becoming a researcher than a doctor. Is really where you landed.
That's right. Exactly.
That's awesome. So tell us now. So you graduate, you get
Yeah.
your PhD. You're now the scientist who's doing all this cool work with
Hmm.
bacteria and stuff and studying viruses, bacteria, and how it's connected to illness. What are you doing in your career at this time, and how's your health?
It was okay. And I started to work at a different nonprofit that I worked at for many years, looking at the role of infections in autoimmune diseases. And it did a lot of building these technologies to better identify organisms. There was a big component of that. A lot of looking and beginning to scope out therapies that might support the immune system instead of suppress it in the treatment of persistent infection and chronic disease, which is still just maybe the thing that I think is most needed. It was the beginning of working with a team and a nonprofit that looked for a paradigm shift in medicine where if we're able to understand that infection is at the root cause often of processes even that are autoimmune, um, then we don't always want to shut down the immune system as treatment. We might even want to support it. And that's still a lot of what we work on at Polybio today. For example, in long COVID, in people who have not cleared the SARS-CoV-2 virus, we helped to start the first trial. It's at UCSF of a drug that activates natural killer cell activity in those patients to bring the immune system up to better fight it off. And that's what I think the future is. And it's a focus that I've had for now many years that is still an ongoing thing.
Yeah, I'm 100% with you. Help us identify this at a simple level, because we hear this all
Yeah.
the time. And how I understand it is, when somebody is really unwell, especially if they have autoimmune like conditions, they're
Yeah.
given an immune suppressor, a
Yeah.
steroid, something that's going to suppress the immune system because they're extremely inflamed, they're exhibiting these autoimmune like conditions, and they go on an immune suppressor.
Right.
But in many of those cases, there is likely a microbial root cause, and by doing that, you might be alleviating the symptoms because you're suppressing the immune system. The immune response is really what's responsible for your symptoms. The immune system responding to the bad thing, the bacteria, is causing your symptoms. But although you might not be having symptoms from the immune suppression, you're letting the microbes flourish and grow and become even more prevalent in your body, which means you're getting even more sick despite the fact that you're having less symptoms. Is that an accurate assessment? You can tell me
Yes.
I'm totally wrong.
No, that's exactly correct in the way that I see it. I mean, what and I think the reason for that in terms of understanding mechanism is that one of the biggest drivers of autoimmunity that's connected to infection is molecular mimicry. It doesn't underlie every form of autoimmunity, but it's increasingly understood. For example, in MS, there's now a couple examples or one example where a pathogen, let's say a virus or a bacteria, makes a protein, and that protein is very similar in size and shape to a human protein or receptor. So when the immune system targets that pathogen protein, it cross-reacts and targets the human, protein or receptor at the same time. And
Wow.
it's kind of like if you were trying to target an army and your enemy was dressed in red, but there were also civilians in the background dressed in red, and you just got everyone, right?
Yeah.
It's that, and so much of science only looks at that second part where the human part, the human protein or receptor is targeted, and they don't know that the original firing was on that pathogen protein in the first place, if this makes sense. And
It
so,
does.
yeah,
So, do you mind if I just, if my own brain to make sure I understand
yeah,
that? I think you're saying that, so Lyme bacteria, for example, likely can create something in the body that resembles a human real thing that's a good, healthy part of the human experience,
exactly.
but it really is the Lyme bacteria. So it looks like it's real
Mhm.
and human, but it's really a bad thing, Lyme disease in the body. And the immune system gets confused and says, I'm going to go kill that Lyme disease, which looks very much like the human body. And oh, by the way, because it's still similar, I'm also going to kill the good stuff and attack the human body
Yeah,
component as well, which creates that autoimmune like, paradigm,
yeah.
which then makes doctors put people on immune suppressors, but then that bacteria just
Mhm.
flourishes. So is this an evolution, do you think, that bacteria has evolved in our generation, or has it always been doing that and we just discovered that that's what it's doing to kind of hide from the body and for to survive?
That's a great question. And so, you have to wonder, why do pathogens even exist and why do they want to hurt us? But they are constantly in it for themselves, right? And it's just the case. And so any time that an infection that gets into humans can evolve to do something that hurts the host more, it generally will. And so it does seem that the pathogens that make proteins that are similar in size and shape to important human structures and receptors that can lead to autoimmune type mechanisms, like I described, this molecular mimicry, where targeting of the pathogen protein can also lead to targeting of self, are the ones that survive better, right? And so it's kind of like evolution has enhanced for pathogens whose proteins are bigger mimics to human structures and receptors and can cause more damage that way. There's lots of examples of, there's even a family of viruses that was shown to make proteins that are very similar in size and shape to insulin, human
Right.
insulin. And it's like, you know, once you're a virus and you can do that, you can cause a lot of dysregulation because now the immune system starts to confuse what's the viral insulin protein like protein with the human, and it's just another way of throwing off your host. So yes, generally speaking.
Yeah. So a follow-up question to that is we've had other researchers on, like Dr. Tal, for example, who told us that we're not sure why, but we do know that certain immune systems respond differently to the same thing. So
Mhm.
my immune system may see that molecular mimicry and go, up, I'm gonna go crazy and just carpet bomb even Matt's good tissue, right? And good cells. Whereas yours might be less chaotic and it might just have some collateral damage, but not nearly as much. So you'll become less sick than I would. And we're trying to figure out why the immune system in Matt is sort of more confused in the immune system in Dr.
Mhm.
Proal. Is that something you're seeing too, where it's not just the one component, it's kind of multifaceted, where the immune responses vary from patient to patient. Plus, we're getting confused by the bacteria and attacking good and bad.
Yeah, I disagree with her a little bit. I don't think that the what I think is one of the bigger variables in how a person responds to an infection is the other infections or exposures that the person also has. So, is in other words, if someone already has Bartonella, and that at most, like most organisms, it's downregulating or there's some, it will impact the immune system somewhat because all organisms do that. Then when they get a next infection, let's say they get COVID, they may respond to COVID with a more hyperinflammatory or dysregulated response than someone who didn't, right? So,
Yeah.
and exposures can also fit into that. So if someone's been exposed to mold and they have a big problem with mycotoxins, their immune system may be dampened or dysregulated in a way where they can't handle the next infection as much, and that becomes more of a problem as well, and they cannot clear that infection, and then that infection persists and downregulates or hyperactivates components of the immune response. And so I think that And sometimes I've actually referred to that as successive infection, where each infection or exposure, honestly, or even sometimes injury that a person gets can dysregulate or throw off the immune system in a way that makes handling the next one more difficult. And it becomes somewhat cumulative in a person, these different hits, sometimes we call them multiple hits, that
Yep.
can become part of a person's case that sort of build up to the fact that when they get the latest infection, they struggle and have an immune response to it that's aberrant. It doesn't mean that there couldn't be something wrong with someone's immune system too, and there are human genetics and different factors that can play into, different forms of immune responses in different people. But I do think it's those other infections and exposures that can be the biggest players in the way immune system responds to an infection in a lot of people's cases.
Right. So, for example, Matt Sabatello gets infected with the same exact type of Lyme disease as Dr. Proal. Matt
Hmm.
gets really sick. Dr. Proal, much less sick. Why? You're saying that the biggest factor you believe is because Matt had other infections and other exposures to other pathogens where he's already weakened because he's had Bartonella, he's had other diseases, he's had
Yeah.
other parasites. So now my immune system is just destroyed and it's just going haywire. All these different things that I'm
Yeah.
exposed to. I'm all beat up. I'm tired. I've been fighting nonstop, and I don't know what the frick to do. So I'm gonna go left and right and haywire and mount all these antibodies. Whereas your body isn't as overwhelmed, and therefore you're mounting a more targeted response to the Lyme bacteria, the same bacteria I
Mhm.
was exposed to, and you're less
Exactly.
sick because of it. Is really your position here, right?
Yeah, I think that's a lot of what plays into it. There could be some differences in the immune system, but I think that's in a lot of the scientific community, I think that's unrecognized. The successive nature of how infections, exposures, and injuries can build up to amplify or change the immune system and the way it deals with subsequent hits, or
Yeah.
infections. I think that that's the biggest part of the picture for a lot
How
of
do you
other cases we see.
how do you recommend we handle that? Because to your previous point, we don't know yet all of the pathogens that exist. So it's impossible to target them. It's impossible to know
Mhm.
what Matt's been exposed to versus what Dr. Peral's been exposed to. So how do you reduce the risk of the next infection causing you to be chronically ill or debilitated when you don't even
Yeah.
know how to run tests for all the things that are out there? And you don't want to just carpet bomb your system with a broad spectrum
Exactly.
antibiotic because there's negatives to that as well. So how are you approaching this from a treatment standpoint when we don't have enough data or science yet to really help people?
Yeah, I do think it's a difficult situation now with this, but at least that's why our foundation and our research programs are focusing on two main things: better diagnostics,
Yeah.
so that you could have a better idea of which infections are in you, and even in some cases what they're doing, not just if they're there, but if they're actually skewing the immune system or if they're making a protein that we know can throw off the immune system, right? So markers of not just presence, but activity, those
Hmm.
kinds of diagnostics, which is possible with enough
Are there
innovation.
tests? I'm sorry to interrupt, Dr. Pearl. Are there tests like that today? Because I'm not aware of tests that identify is it there from a
Yeah.
bacterial standpoint, but also what is it doing? Do we have tests like that yet, or is that something
Not
you're still
yet.
okay?
It's what we're working to develop because, for example, you can, there are labs now, and again, none of this is commercialized yet, but could be, where you can take a T cell and sequence the fact that it's still responding to a protein made by, let's say, Borrelia. And
Hmm.
then you can say, not only does that mean that the protein must still be there, created, in Borrelia, but also we know that it's making the person's T cells respond to it. And you can measure even relate a couple of related inflammatory parameters that could show you then that that T cell is exhausted, for example. When the T cells are exhausted, they express a specific marker called PD-1. You can measure that and you can say, this person has Borrelia, their T cells are exhausted, and you can gain more information from that type of testing, right? So that's the kind of diagnostic that would be possible that would give you more info. Because if you were then in a clinic, you would say, maybe in this person, a mix of a treatment that targets Borrelia directly, but also something that supports T cell activity to bring back to remove the exhausted state would be optimal for their treatment.
Hmm.
And that's
Yeah, that's fascinating. I
it's a,
mean, let's talk about the other side of this, which is there are other articles and studies that have come out over the past 5 years that talk about, and this is the age-old debate. Is it an active infection or is it an autoimmune response after the infection,
Mhm.
right? Post-infection versus chronic recurring infection. And, you know, I don't know, different people have different perspectives, but there's one study that came out that talks about the remnants after an
Mhm.
infection is cleared trigger an immune response, but it's not the live or active or living bacteria. It's the traces or the residual or the aftermath of the bacteria. It leaves little proteins, I guess, in the body, in the tissue, in the blood. And the immune system responds to that, causing an inflammatory response, causing symptoms. What is your view towards that? Do you think it's exclusively a chronic infection, or do you think it's a part of it where the dead bacteria is shedding things, causing a response as well?
Yeah. I think we need more research to definitively prove what is going on. But look, I'll use long COVID again as just an example because we, as our foundation, we built the first ever research program on long COVID. Just the SARS-CoV-2 virus emerged. And what we already knew because of the Lyme space was that it was going to be very easy for people to say, like, in some long COVID patients' blood early on, we found circulating spike protein. And you could just say, well, maybe they just have the protein and not the virus in them anymore. You know, that it's a little bit of a similar scenario. But what we knew is that the virus, the itself was unlikely to be in the blood. So we started a program where we had teams collect tissue from the gut, from the lymph nodes. Patients would undergo, and patients have been so awesome in these research studies, undergo an endoscopy procedure to get tissue from the lining of the gut, or a lymph node biopsy to get tissue from the lymph nodes, or even in some cases from the thigh where there's peripheral nerves, right? And in those cases, especially in the tissue of the gut, there's still teams who have found persistent SARS-CoV-2 RNA in the virus there, even though it's not in the blood,
Hmm.
right? And so the thing is, if we hadn't done those experiments, let's say those groups had never been supported and had never collected those samples, the long COVID field would just think that SARS-CoV-2 couldn't persist and that only spike protein was sometimes left over because that's the only research that had been done. So when you go into the chronic Lyme space, there are very few teams there's no tissue biopsy, and we're building this out now from our foundation, but there are very few groups that have actually gone and said, let's get tissue from the joint of the knee and the synovium from people with, chronic Lyme and joint symptoms, or let's get a lymph node sample from these patients and let's look for Borrelia there, and not just, sort of assume that it's just because that's not in other fluids or something that it's not there. And I think in the case of what you're talking about is peptidoglycan, created by Borrelia, where it's a component of the bacterial cell wall. And there have been some studies, especially in Lyme arthritis patients, where they've looked in the joint fluid, the fluid of the joints, and they've found peptidoglycan. And what they say is that it's just the peptidoglycan there, causing an inflammatory reaction. And this could be the case in some people, but based on the work we've done in long COVID, I think we need to get more tissue from the joint. Like, they're looking at the accompanying fluid and they're not looking at the accompanying tissue. And that's where the signal might be where you'd find the Borrelia. And so until that study is done, which we're working to do, and you do that with a couple other tissue types, it's it's hard to know if it's just the peptidoglycan or if you just didn't look for the organism itself in the right place. And also sometimes with the right methods, because depending on, sometimes when organisms persist, there's very little of them in a sample. And Borrelia is famous for this. Like a few little Borrelias can drive a really insane inflammatory response. So you have to have very sensitive methods. Like when you're looking in tissue, you have to be able to amplify like a small amount of the genetic material. So there's a lot of things that can go wrong, in other words, in not finding persistent Borrelia because you might just not have a deep enough tissue sample to find it. You might have a team that is looking for it, but just doesn't have method sensitive enough to find a small amount, So the whole thing is that there has to be a lot more, of these projects in the space right now that get these tissue samples and that really innovate the methods that are most sensitive to identify Borrelia. And that's going to help us kind of answer this question more, in my mind.
I
Yeah.
love that answer. I'm learning so much from you. So a follow-up, because you're provoking so much thought, is you talked about earlier, it's the successive infection, right, approach, which is why you believe some people get more sick than others, which completely makes sense. But how would you respond to the stories we hear all the time? Hey, Matt was bit by a couple of ticks. He got chronically ill. But there are people that go hunting, live in the woods, and pull thousands of ticks off them every single season, but never have been sick. They live healthy lives, and they've never had a tick-borne infection that they know of. To me, that's far more successive infections because they've probably been infected. I mean, from a probability standpoint, they had to be infected numerous times with various tick-borne illnesses if they're getting bit by thousands of ticks a year,
Yeah.
which you'd think they'd get really sick from one of them and be debilitated. But how would you approach that? Why are those
Hmm.
people just not getting sick if it's really about the successive infection model?
That's a great question. There's a couple variables there, which, one, and this also requires a lot more research, but it's important to do, is that, you know, sometimes these infections can be passed from mother to child or even potentially from father to child, especially from mother to child in the womb. And I think that there needs to be a lot more research on people who might inherit microbial contributions to already having some low level of these organisms that sets them up for early immune dysregulation so that if they're reinfected, it becomes even more of a problem at a later time. And that's not studied right now, and it needs to be studied a lot more. So in the same way that our genes are inherited from our parents, we need to look a lot more of the way that microbes are inherited from our parents as well, and kind of set up our immune systems to handle what happens later in life with other infections.
Yeah.
And that's a base level factor.
It's interesting because that's something an observation we've, after speaking to
Hmm.
thousands of people in this community,
Yeah.
our observation has been that the child population, meaning the people who got sick at an early age or had congenital Lyme,
Yes.
when they're still not fully developed adults, generally get even more sick, but more they
Exactly.
have more neuropsychological symptoms, more, pandas, things like that. And we've always wondered why do children respond differently to tick-borne infections than adults? And do you think it's because maybe their immune systems aren't fully developed? Do you think there's a multiple facets to that? Like if you're getting these infections at birth from your parent, from your mother, are
Yeah.
you less prepared to manage it because you're still developing body and
Exactly.
immune system, and do you think that's a contributing factor?
I do. And again, we need a lot more research, but just speculating on it, any time where the infant's developing with an infection present, that cannot be good. It probably allows the infection to get into a lot more sites. And I think, another factor that becomes in my mind, whether it's in childhood cases or even in adults, is the degree to which the organism gets into the central nervous system. It depends, but once the organism is in the central nervous system, it could definitely cause a lot more problems. I think that especially because let's say that the organism does make it into the brain. The brain has immune cells called microglia, and those cells drive neuroinflammation. And a little bit of organism can start to activate those microglial cells in a way that primes them. And what that means is that now they're in a state where when they encounter the next infection or the next exposure, they respond in a bigger, like they respond in sort of an exaggerated way. So that's a way that, and that's in the brain, which is a very kind of contained space. There's not that much room. Like, let's say, you hurt your arm and it swells up, right, with a bunch of inflammation. You can kind of handle that in your arm because it's of the area. In your brain, there's such little space in the central nervous system, for inflammation that can be kind of cumulative like that to exist, that a little bit of like microglial priming from one or two different infections or exposures that start to compound this can become a huge issue. And now the person gets the next infection, it becomes like utterly magnified because the system and the central nervous system is just already kind of, overly primed. So I do think that sometimes this is the location of the infection becomes another variable in people, which could just be luck in some cases. But if it gets into the brain or spinal cord, where in case people, especially if they have meningitis or really have, an instance where, and for example, one of the things we look at a little bit is how some organisms can even travel the vagus nerve, maybe. Like if the pathogens can over time travel nerves and get more into the brain, that's going to be a worse scenario for people when they get the next one as well. And
Hmm.
that's a little bit of sometimes why we see. and it just depends on cases and again needs more research. But, a lot of times you sometimes will see that someone who has more problems with infection also did have a history of injury. Like sometimes patients will have a history of concussion or other injuries that go along with the fact that they struggle with an injury. And it's likely because those injuries are already priming the brain's immune system. And now when they get an infection, you're in way worse shape as well. So it can be like some of those things that converge.
Yeah, I'll tell you that observation we made pretty quickly on this podcast. I remember saying to Rich one day, why do so many of our guests tell us they've had so many concussions? And I said, I don't understand. Is it because they're neurologically impaired and they're off balance and they're falling and banging their heads and getting concussions? Or was the concussion a precursor to Lyme and it made them more vulnerable? We've asked other doctors and researchers, and they're with you, where they think that, these injuries, these concussions before Lyme make them more susceptible and vulnerable. And it might be, a common trait where, physical injury, pre-existing illnesses, to your point, the successive infection model, right? All that kind of stuff makes you more vulnerable to be really sick when you get infected again and cripple us.
Yeah.
So I think there's a lot of anecdotal evidence there and obviously scientific evidence on your side, which is interesting to hear that kind of come together.
Yeah. And another big one, honestly, is the microbiome in people. So, we do have these trillions of interacting bacteria, fungi, these communities in us, right? And, under conditions where the immune system is strong and in good shape, those communities are, in a state of balance. But if the immune system starts to weaken or there's, and this could be so many things. It could be someone exposed to mold or microplastics or too many, that those organisms can basically move to a state called dysbiosis, where the bad bacteria start to take over and the whole ecosystem of in the gut, but also in the mouth, and increasingly understood in other sites like the lungs or other body sites, starts to skew towards a dysfunctional, pro-inflammatory, community. And that doesn't help anyone out at all either, because what can happen, and that's where you start to see people, even practitioners try to address leaky gut or things, is if, let's say, just the gut microbiome alone, you have bad bacteria overgrowing, you have, organisms that are not good becoming more predominant, the immune system tries to recognize that there's inflammation and that wears down the lining of the gut. That can happen in the mouth. It can wear down the lining of the mouth. And that means some of those bad organisms start to seep into the blood, where they can, they'll definitely flare them. Once they're in the blood, the immune system will hyperactivate. That becomes a huge thing. And then when you get another infection, your immune system is already dealing with some of those, translocated gut or oral bacteria or something like that. And that is a problem for you as well, right?
Yeah.
And so like the whole eco state of the other organisms in your body is a whole other variable, where some people are in good shape on that front and other people definitely are not. And that's another thing that can, and even sometimes like the composition of the microbiome. And a clear example is there's a vaginal microbiome. And if it's there's research that shows that if it's in a balanced state, a woman is less likely to acquire HIV from a partner. And if it's in a dysbiotic or, skewed state, they're much more likely to get HIV. And so the way that the microbiome behaves sort of enables if another pathogen can kind of get into that space and set up shop or not. And so it's a whole other variable that matters too.
So let's break this down a little bit more. So the microbiome is basically you're born with it. It's part of you, right? So the microbiome, you have good microbes and bad microbes, and they live together and in balance. But sometimes it becomes out of balance, which is this dysbiosis you're referring to. But that's different than you contracting an infection like Lyme from a tick bite, or you contracting a virus from somebody sneezing on you, right? Those are more acquired illnesses versus your balanced microbiome, which makes up who you are as a human being, right? But I think you're saying that the microbiome, if it's out of balance and there's more bad than good, can make you more susceptible, like the example of HIV, to become even more either get infected altogether or become more sick from an infection if your microbiome is out of balance, is what you're saying, correct?
Yeah, exactly.
Okay.
Yeah.
So, on that note, I feel like that this conversation we've had a lot with a lot of other doctors and researchers, and I'm curious, some of the things we talked about are like fecal implants, right? So people will go and it's either through a capsule or through like a little an implant, they put it up into their colon, I guess it is, and they'll get the they'll go out and shop online, or they go, and I don't know how the whole thing works. We've had heard different stories from different
Yeah.
people, but they get a pretty diverse set of microbes from somebody else's, feces, basically, and they take that to replenish their gut microbiome to have a healthier balance to be more resilient to existing infections and prevent future infections. What are your views towards that?
I think it's reasonable. I I think and there's groups who are literally doing fecal transplants, and there's some really good data on fecal transplants and ulcerative colitis, for example. I actually think there's even one or two clinical trials that showed that patients with ulcerative colitis who are essentially given, yes, a new microbiome based on a fecal transplant can sometimes really improve their symptoms as opposed to the really strong immunosuppressive drugs they're otherwise given, right? And that's an example where you have a gut-based disease that's increasingly understood to be tied to these organisms. And at that point, I'm like, yeah, if you can switch them up, I think this makes sense. I do think the donor matters. You have to make sure that the donor doesn't carry something bad in the, fecal transplant matter that you would get. And that's a whole thing. You have to really try to screen the donor, and that's a thing. But I definitely think that more work and more research and more innovation on that front is a path. I think that's totally reasonable. There are also companies that are trying to make, you know, really good microbiome-based therapeutics that are somewhat, derived from, like, they'll take like top athletes who function really well and kind of take their, like, isolate what they think are the top microbes that help people out and create that into a more artificial but hopefully living supplement. And I think that's a really cool area as well. Like, the dream would be if you didn't literally have to use someone else's fecal material and you could use something really well recreated. Although maybe it will always be that just because the thing is the fecal sample will have organisms, but also immune factors and other components to it as well that, I don't know, might be hard for humans to recreate. But I
Yeah.
definitely think that what I'm going to call microbiome changing therapeutics is a very important area that needs to keep moving forward.
Have you read a study from a few years ago that talked about they studied chronic Lyme patients and found an increase of particular two microbes in the microbiome of chronic Lyme patients and a decrease of another that
Mm.
were consistent across all chronic Lyme patients. And I can't remember what the three things were, but there was an increase of two and a decrease of one that were consistent across the board for chronic Lyme patients. Do you know that study, and what are your thoughts about it?
I think I know it. Um, I think I know it. I guess I'd like to see the results replicated in a couple other groups. The thing that makes microbiome research hard is that there's no core gut microbiome. So depending on where, which country people are from and what area of the globe they're in and what their diets are and just familial transmission over many years, like you could be healthy. One person could be healthy and have a totally different composition of microbes in their gut than the next healthy person. So there isn't this base comparison where we're like, oh, this group is always good, right? So it can make some of these microbiome studies a little harder because you'll see changes in this species or that one, but sometimes when you go to like a new group of chronic Lyme patients or something, it doesn't replicate. But I don't know. I think overall, so my main thing was like I wouldn't rush out and get some kind of probiotic based on those results, like in some big way. But I do think that continued work to see if there is a type of bacteria or, family of bacteria that could be depleted more in people who are more susceptible to Lyme or something is totally reasonable. Like, yes, I mean, we should be always looking for those types of connections. Yes.
Yeah, and I found the article is Kim Lewis from Northwestern,
Yeah.
and
All
it was
right. Makes sense.
specific. He said that there's an abundance of a bacteria called Blautia,
Hmm.
Blautia, and a suppression of a type of bacteria called bacterioids,
Yeah.
and that was pretty consistent across all chronic Lyme patients. So just interesting there.
And those bacterioids are kind of seen like coming up and down in a couple of different chronic diseases. So there are especially, families or classes of bacteria that seem like either not good or bad players across human health. And so there might even be um, like, for example, bacteria that can produce short chain fatty acids are actually good. You want short chain fatty acids to help with the certain types of signaling and certain kinds of food breakdown. So there's some probiotics that go in that direction. So I don't know. I think it's a totally reasonable, a really good area of exploration. What I don't think though is sometimes people will like as much as I think microbiome and chronic Lyme and Lyme related research is a really great area, sometimes people will use the microbiome as an excuse not to ever treat Lyme. And I think those people are going too far. Like I've had people say, like, oh, this person got a tick bite and we don't know if we should give him doxycycline because it might impact the microbiome. And at that point, in my mind, it's like, look, like I'm more worried about the Lyme that they might not clear or might have problems with than I am with microbiome. There's a lot we can do with microbiome, and honestly, there's a lot of studies that show microbiome can be kind of resilient. So I like the whole area of microbiome research while not thinking it's enough of a reason to not use antibiotics when they're needed.
Right. But let's talk a little bit more about that because
Yeah.
we've heard from other people on this podcast that the third world countries don't have ulcerative colitis, right? They call it a first world problem. And they say that it's a variety of factors. They say that third world countries, you're exposed to more of nature, to dirt, to mud, to just the out,
Mm.
the real world. Whereas we're sanitized in first world countries like America. So we have these first world diseases like you see. And
Mm.
beyond that, we are more treated with antibiotics compared to third world countries, and the antibiotics are destroying our gut microbiome from a diversity standpoint. So the sanitation or the sanitized lifestyle of living indoors and the less diverse microbiome from antibiotic overuse is what makes us more susceptible to these first world diseases. Do you think it's a sound argument, or do you think it's flawed?
Uh, I don't love it. It's called the hygiene hypothesis often. I there I think elements of it make sense. But first of all, you have to understand that we are not living in a sterile environment. I think we're just if you're in a city or something, you are exposed to different microbes than a person who might be living in a rural community. But believe me, if you get on a New York City subway
Yeah.
and you touch a pole, like there's the number of organisms you will pick up are huge. Or like there was a group that did a sequencing study of the average sponge on a kitchen sink, and I mean, there's trillions of so no one's sterile. That's the wrong argument. You could say that you think that urban bacteria are less good than rural bacteria or something like that, and that might make a little bit more sense. But no one's sterile. That's mis. I don't think that's correct. But I think then, okay, I get it. Like maybe bacteria that are in the soil, and like there's soil-based probiotics. I think this is reasonable. But people also can take that a little far in my mind, where they're like, and every kid should have a dog, and you should all like, and it's like, I get that, but be aware that dogs can also carry fleas, which can carry Bartonella, and all like, it's like the argument doesn't include the negative things that could be
Yeah.
part of all these exposures. So I think there's a middle ground where you want to be exposed to organisms, but you just have to think intelligently about like you're not like, oh, I wanted every animal should be in my contact now. Like in fact, veterinarians are some of the people who have the most trouble with chronic tick-borne vector-borne illness. If you ever look at Ed Breischwitz's work, right? He's like
Yeah.
doing case series on veterinarians. So the idea that, a lot of animals exposure is not always great. So I think that overall it's not my biggest worry. I don't think we're too sterile. That being said, like, do we want to think about making sure that our microbiomes are in good shape? Yes. But the other thing that I think is not factored in enough is, when people say, oh, we're using so many antibiotics, antibiotics are overused. But a lot of like, before we totally don't use antibiotics in the treatment of important human infections, consider how much we use them in our livestock. Consider how much a huge amount of antibiotic resistance that is driving there's major problems with antibiotic resistance. It's coming from farms and animal where we are just making animals guzzle antibiotics. We need to stop those practices and a lot of real misuse of antibiotics before taking them away from people who have infections that need to be treated. So there's also, when you get into these antibiotic resistance discussions, a lot of factors of where that's coming from. And I think some of those are, depends, a little skewed. And I think also the last factor is the immune system. A person's immune system is a huge factor in how healthy their microbiome is. Because when the bad bacteria try to overgrow in someone's microbiome, if their immune system's strong, it will shut it down. It's like, no, it'll come in and be like, no, you can't overgrow. Like you're going back down. You know what I mean? So a huge variable is how strong people's immune systems are. And that is, I think, a factor in the US that's a problem. I mean, we are exposed to so many factors that, from pollution and, toxins and all of these things and food additives, all of these other things that are part of first world living that are not great for the immune system in terms of the exposome that is very bad in a lot of our lives are other factors that might not be happening in a rural community in a different country, which means they might have less exposure problems and have stronger immune systems to keep their microbiomes in better shape as well. So we have
Ooh.
to factor
Oh,
that
I
into
love that. You just like literally turned it upside down on that.
a little bit, yeah.
Oh, I
I
like
mean,
that.
I don't
Sorry
know.
to interrupt. I just really liked what you said. So the bring us back to where you were, because I took us on a big
Yeah.
tangent there. But how did you end up now? You're post medical school, you're doing all this research, 2005, you're scraping away the human side of it, looking at all the microbe side of it. How do you go from there in 2005 to, I think it was
Yeah.
2018, where you co-founded Polybio and all of your work now with the Cohen Center for Recovery from Complex Chronic Illness?
Yeah, so I worked with that other nonprofit for a long time. And then I actually, around maybe 2015, I took a break. And I lived near my twin sister. And well, first of all, I did take a little extra breaks for some more health related things. And I did end up actually late in the game testing positive for Bartonella, Babesia, a couple infections, and I treated those. So, like, in a story of, these things being ignored, it might be that even one of those infections I got as a kid was a mistake bite.
Hmm.
So
How did you
that,
treat those two infections?
yeah, I did tefenoquine for Babesia and a couple antibiotic protocols for the other ones on the most part. Yeah. So, so did those and kind of addressed that a little bit more, and then worked basically. My sister and her husband were starting a sports analytics company for water polo. It's just kind of random. And I was living nearby them. And I took a break to help them get the company off the ground. And it was like a typical startup. We were building pitch decks and we were this and everything. And that's the time when I started to look at other foundations in the infection associated chronic disease space and think, we've got to innovate the work that's being done here. And I want to start a foundation. But if I do that, I don't want to start a foundation that just raises for the cure or for something ambiguous, right? I don't want to raise money for like chronic Lyme. I actually want to show people what we're going to do, like build the projects first and then raise the funds to make them happen. And so I took their pitch deck model and all of this startup model and created Polybio as a foundation where what we do is, yeah, we build our programs first. We're like, okay, we're going to do a diagnostics program. We're going to get this tissue. We're going to get these samples. These teams are going to analyze it. This is how it's going to work. And we build those out. And then we go to private donors and we're like, hey, if you support this, your return on investment might not be like investment or, profit like a startup. Your return on investment is knowledge. And so that's kind of what helped me build Polybio with a model where we do a lot of collaborative work and we use our scientific expertise to build out programs, which became a big thing of how we are operated. And then I kind of, at the time before Polybio, I had a blog called, I think, Microbe Minded, where I started to write more about infection. I started to do some videos, and then I just kind of gradually turned that into our foundation.
Hmm. And how did that bridge your work with Mount Sinai at the Cohen Center for Recovery?
Yeah, so Polybio was going strong. And when long COVID became a huge thing, and we started doing a lot of work on the persistence of SARS-CoV-2 in tissue, which is a big factor, that's where David Petrino, who's my colleague at the center, was wanted to start a long COVID clinic and was doing, I mean, it's a really good job already, but he he did recognize he didn't have a microbiology or virology background. And so I'm flattered he came to me and was like, Amy, I really want you to be the scientific director of this clinic. You've spent so much time in this microbe space, which is just true, that I think you make sense as the scientific director of this site. And at first I was like, David, you're crazy. I already run a whole foundation. I cannot do that. And he was like, just try, just try. So he lured me in. And then the truth is, it's a great team. And quickly, what convinced me to do it is, like you were alluding to before, I want to know what's happening with patients in the clinic. I want to know what patients are saying, what patients are reporting they're feeling. I want to know exactly what gaps exist when a patient comes in for care. And you're right, we don't have that test, or we don't know what to say when that happens, right? Because then that knowledge goes directly back to informing the research that we need to do to fill those gaps. And so it's a really actually good dynamic where I always know what's up on the patient front and can feed that back into the programs that we build. So it actually ended up working out really well.
Yeah, and I think it's a really good model to have the research plus the clinical side of it, the patient,
Yeah.
the human element, seeing patients, and then having the researcher mind, like you said, like when you're like, hey, they're walking in to see the clinic to get treated for chronic Lyme, and I'm thinking, what could I
Yeah,
do with this patient group to further our research? And you're
exactly.
coming up with ideas on the fly, and then
Exactly.
conducting these research studies in collaboration with the actual doctors who are treating Lyme patients, chronic Lyme
Mhm.
patients in real time, which I think is a really good
Exactly.
model.
Yep.
So what have you
Yeah.
learned? What have you learned from doing that? Because, in all your work through Polybio plus the Cohen Center for Recovery, I think you've had to come to a lot of conclusions that were probably new conclusions because of this mass quantity of research and patient experience that you're having. So tell us what you've
Mhm.
learned so far.
The two biggest things I learned in just the side of the research is that if you don't collect tissue samples from patients with an infection-associated chronic disease, you will never know what's actually wrong with people. To the point where we and the teams who most understand long COVID now start their talks by saying this is a tissue-based disease. This virus infects every major human tissue type. If we only studied a person's blood, we will never know what's wrong, throughout the body and the brain. And to do that, once you have that understanding that to the point where our slogan is actually the issue isn't tissue. Like I made
I
that
love
up,
that.
but that's really how I feel. The issue isn't tissue. And so once you know that, you can do some really innovate research because not only can you do these tissue biopsy studies that can be hugely illuminative in what's really happening in a person's body, but we can iterate imaging. So, we have a project, for example, at UCSF where the team puts a patient with long COVID, in this case, into a full body scanner. And then they're working on like a radioligand kind of tagging a molecule that can identify the SARS-CoV-2 spike protein throughout the body. This is still an ongoing study, but technically they can image the SARS-CoV-2 spike protein throughout a person's body using imaging methods because they've tagged it the person in a safe way. They take a molecule that binds to the spike protein, put a little fluorescent molecule on it, let it go throughout the person. If it finds spike, it binds it. And then the person is in an imaging scanner, and the scanner can light that up and map it, which is so awesome.
That
They've
is so
done it. This
cool.
is a group that did it with HIV. They have a paper out where they just map in an HIV patient using the same method of tagging a molecule that fluoresces when it binds to HIV. They map HIV reservoirs throughout their body and brain. And so that's infectious disease molecular imaging. It's such a cool area. We're working on innovating that. And so I think, but it's that basic understanding of being like, I have to know what's happening in a person's brain and in their nervous system. And I'm going to push the boundaries of technology until I know that. That's the one of the
Yeah.
biggest things that I think is necessary.
Real quick
And
to
yeah,
interrupt. Sorry to interrupt real quick, but can that tracer that can get into the brain through the blood-brain barrier and deep into tissue? So if the infection is hidden deep in an
yeah.
organ or in a tissue or in the brain, it can get there, those tracers. It's going to show the full body experience.
It depends on the tracer. It's a great question. That it's a whole field to design tracers that are capable of crossing the blood-brain barrier. Sometimes someone has a really good tracer, but it doesn't do that. So they will only, they'll have to say, oh, we think we can, identify the organism in the body, but not the brain. But then sometimes, because these teams have chemists on them, they have they'll be like, we're going to fix it. There's even groups that there's a couple of research teams that have like a helmet that can temporarily open up the blood-brain barrier quickly. They do this in cancers, so that it could get in quickly for the study, and that like it's
That's wild.
definitely a little wild, but
Yeah.
it's an issue that is certainly thought about, right? Where you have to consider you're very smart to think that because a lot of people honestly don't. They're like assume that the ligand gets into the brain and it very well might not, right? But that's a whole area of research.
Hmm.
So yeah, there's lots of different ways you can even tag like use CRISPR based methods, which can kind of like identify our like a dream, which is literally something that we're trying to build slowly, is that you could use an imaging tracer to identify like a persistent, this, let's say virus, the backbone. And then if the tracer identifies it, it can also deliver a treatment to break up the RNA. It's called a
Wow.
theranostic, where
Huh.
the diagnostic also delivers the treatment, a theranostic.
That's
Really
insane.
cool, right?
Oh
So
man.
imagine that. And that's imagine a future where you could go in, be like, okay, do I have Borrelia in me? They'd be like, get in the scanner. They'd safely inject you with a radioligand. It would spread throughout your system. The monitor would light up where the Borrelia is, and then they'd, inject the thing to break it up. That's the dream.
Doc is pulling it up. Do you know Dr. I'm gonna butcher his name, Jayakumar Rahadas from Stanford? Do you know what I'm talking about?
No?
He's a Lyme researcher, and he's working
Okay.
on. So he's a gentleman who discovered various treatments. So he discovered azlocillin. So he was the man behind that just came out last year. Disulfiram he discovered, and now he's
Okay.
working on
That's
another protein that's derived from the bee venom, but it's a
mm,
compound, not actually real bee stings, obviously. So he's done a lot of work in the Lyme community, but he's working on these things as well.
that's good.
And it was a fascinating discussion, but very similar, where he's identifying these compounds that can go and find the
Yes.
Lyme bacteria, but at the same time find it and then kill it. So it's like a very
Yes,
similar
exactly.
idea. You should look him up
Exactly.
because this guy was
Oh, I will.
brilliant from Stanford, and I think the
That's
two of
awesome.
you would be an awesome collaboration team. I mean, brilliant. I'm going to drop his link into
All
the chat.
right. Put in the chat. I will
Yeah.
definitely look him up.
Anyway,
Uh,
I'm sorry to interrupt, but I just think you two would make
no, but
a great combo.
we would. Yeah, I agree. Cool. All right. I'll reach out to him. Yeah.
I interrupted you earlier. You're talking about that was the first thing that you've learned, the first big thing. I think you were about to go on to the second big thing that you learned from both of these experiences.
The other thing is just, it's nerdy, but the methods. I mean, we've spent so much time, let's say, if we're trying to you know, figure out what is non-human in a tissue sample, you can screw that up in so many ways. Like you have to remove the human genetic material first, and that's an art. Like you have to process the tissue and you can over process the sample in a way that breaks the human material up too much, and then you don't get. So the thing is that you, in my mind, it's better to do, and it's actually one of our polybio values as a foundation, it's better to do one study right than 10 sort of right. Because you can confuse a field by just well-intentioned work that just forgot one step. And you really needed to think through the way you extract the protein or the way the sample is spun or things like that. And it just happens more often than people would think. And so it's really important that that's understood when you're doing research.
Yeah, I do want to ask, especially because now of your partner, I think his name was Mike, right? Who's a
Hmm.
neuroscientist.
Yeah.
The brain is fascinating. We've had a lot of Amen Clinic doctors on, fascinating. The head of iLabs now is from Amen Clinics. And they showed us the brain images and it's right on their website, of various diseases through a PET scan and compared to a normal control brain. And it blows my mind to see a chronic Lyme patient's brain, like in a
Yes.
visual way, compared to a normal control brain and how different the brain is. So, what are
Right.
your thoughts on how Lyme impacts the brain, especially with your partner being a neuroscientist and your background being in microbiology? I'm curious to see what your thoughts are in general about neurological Lyme, the brain in Lyme, and the impacts and how hard it is to treat that component of it.
Yeah, I think, like I was saying, I do think that's the biggest challenge is when Lyme gets into the central nervous system. I do think that the that microglial cell activation is definitely part of that's the base of neuroinflammation. So the imaging that Mike does, for example, where he puts people into a brain scanner, it measures those immune cells, microglia that will activate. It measures those. And that's where he can also map like he's not doing chronic Lyme quite yet, but let's say a long COVID person's brain, like they're going to have all this red area where those cells are lit up and inflamed more than a person who doesn't, right? So, but in some cases where the blood-brain barrier is compromised, you actually get T cells or other immune cells trafficking into the brain where they shouldn't. Those will cause more inflammation, and you get, kind of a compounding of inflammation. And I think one of the things that can also, I think that can impact the central nervous system that Mike does study a lot, that is that you don't even have to have an infection that gets into the brain every time to have neural symptoms happen. And one of the reasons why is because of the vagus nerve that I mentioned before. So you have this really important nerve, the vagus nerve, and it innervates every major trunk organ of the body. So it innervates the gut, it innervates the heart, it innervates the lungs. And what happens there is if it senses inflammation, it will convey a signal to the brain that can throw out, like, can skew brain signaling and up inflammation in the brain, on the other side of the nerve. And the nerve innervates the brain at the brain stem, the back of the brain. And that area of the brain has these overlapping nerve bodies that control the ability to move from sitting to standing, or a lot of autonomic function, right? Also pain, nausea, and what's called the sickness behavior response, where, like, let's say you have food poisoning. You know, how do you manifest the symptoms of feeling sick? Well, what happens is the vagus nerve is like senses that there's bad bacteria in your gut from the food poisoning, conveys it to the brain, and it mounts this response to be like, lay down, you're tired, this sucks. Like that's how you get the feeling of, like, when you're sick, that you're sick. So if the vagus nerve, let's say you haven't cleared Borrelia or some other infection, and it's in the heart or the lungs or the gut, that signaling through the vagus nerve can keep stimulating these sickness symptoms and the pain and the autonomic dysfunction, especially, which is really huge in people's cases where they have, like, major issues with autonomic function. So we look at that as well, where you actually, so Mike, for example, has an imaging study where he images the functional connectivity of the vagus nerve in patients. And even in this case, starting with some chronic Lyme patients, to be able to better understand to what degree signaling of that nerve is also dysregulated and even contributing more to neuro symptoms.
That is fascinating too. So you're saying, although Lyme and tick-borne infections can penetrate the brain via the
Hmm.
blood-brain barrier, in other cases it doesn't, but it stimulates neurological symptoms because the Lyme bacteria infects the vagus nerve, which is not in the brain, which then causes the nerves, which stem up to the top of the brain stem, to tell the brain that there's a problem because the Lyme infection is infecting the vagus nerve. And then it triggers a different brain chemistry to cause these neurological symptoms, you're saying, right?
Pretty much. And it could directly infect vagus nerve, which is something we are fascinated about and are doing in some site, or it could just be like in the gut where the vagus nerve is connected to. And all has to happen, or in the heart, for example. And the vagus nerve just can sense that. It doesn't even have to infect the nerve and convey it to the
Oh,
brain.
so it's either the vagus
So,
nerve is infected with the Borrelia,
yeah,
or it's just the vagus nerve is sensing through its, like, spidey
yeah,
webs, right? That the bacteria exists somewhere near the vagus nerve tract
right.
within the body, and then it's telling the brain we got issues, and then it's causing. Now,
Yeah.
I'd be curious: are there different neuropsychiatric symptoms when it's the vagus nerve causing the neurological symptoms versus the actual infection of the brain?
We're not sure yet. The vagus nerve associated symptoms would be the kind of classic ones that we see. Like, you know, when when you have this overlap where patients who are diagnosed with ME/CFS can often have the same, obviously, symptoms, if you're just going off symptoms, as people with long COVID. A lot of those symptoms are those ones that vagus and brainstem control. So the pain, the nausea, the autonomic dysfunction, the malaise, those are some of the key ones that we know are associated with dysregulated vagus nerve. Some of the other symptoms, like, that can be even more strikingly neuropsychiatric, that's still an ongoing area of exploration. I mean, I know that Mike's team with imaging measures the dorsal, like, anterior cingulate cortex. It's a region of the brain that's involved in cognitive control. So they actually have, this is in long COVID, but they're also working to chronic Lyme. A patient would be in the scanner, and they do a cognitive task in the scanner where they, try to count numbers in a row and try to like, do a couple basic numerical exercises, and they image what parts of the brain light up when that happens. And in patients with long COVID, like you'll see more problems with the cognitive control center, which is not surprising. In other words, like what I'm getting at is, and Celsa published this, it's like unpublished. She's presented at some conferences though, like infection possibly of that region of the brain could be more of an issue in people who have like speech recall or problems with number, you know what I mean, or cognitive issues. So there are certainly areas of the brain that, because they are associated with specific, you know, brain functions, could obviously become more of an issue depending on what's infected, right? Like
Yeah.
another thought is like in pandas, the cribriform plate of the nose, like connects to an area of the brain that is associated with OCD type symptoms, right? So that's where it's very interesting in the study of pandas, like any organism that can get like even through the olfactory bulb or up through the cribriform plate of the nose and into the brain would maybe get to a region that would generate more OCD. And that might be one of
Wow.
the reasons why like even a respiratory infection can be part of a skewing or like setting off a pandas case because you're aggravating that circuitry through the cribriform plate. So,
That's wild.
yeah.
What are your thoughts on, because we talked about Dr. McDonald earlier,
Mhm.
many years ago, he made the correlation between dementia, Alzheimer's, things like that. And then he was finding these brain tumors, and you probably read this, where every brain tumor patient had Borrelia in the brain. So I'm curious what your thoughts are when it comes to the brain and Alzheimer's, dementia, MS. There's a lot of certainly studies that are even coming out. I think one just came out like last week that we published on our social media about Alzheimer's and Lyme and that connection with just current 2026 science. But this has been going on for quite a while. So I'm curious what
Yes.
your thoughts are on this, especially with Mike's involvement and all your research that you're doing. If you
Mhm.
really think this is a sound connection.
Oh, yes. In fact, actually, when you're going back through my life and what really interested me, I became most interested in infection and Alzheimer's disease, actually, across this path, because part of the research that I was starting to follow was a team at Harvard Medical School, and they're still there. It's Rudy Tanzi's group. He discovered APOE which is one of the genes that can predispose to Alzheimer's, but his lab also started working with a guy named Rob Moyer, who did early work to show that the amyloid plaque that accumulates in the Alzheimer's brain might actually have a function. It might not just be like a pathological plaque that accumulates for no reason. And what he studied is that it was part of the basic innate immune response that forms in response to infection. And so he's shown they've done in a neuron dish model in mice, they can put a herpes virus into a brain model and you can watch the Alzheimer's plaque form around that virus. Then they did it with fungi. They've shown it with bacteria. So it's a type conserved part of the immune system where the plaque would form in response to any infection, which of course means that it could form in response to Lyme or Bartonella or any bacteria. And I think that is a paradigm shift. They even put out a paper recently showing that the tau tangles that develop associated with Alzheimer's plaque can also be formed by viruses or other. I mean, like the whole pathological nature of Alzheimer's could have infection at the root of it, and this would just make sense in many ways. This is why it's always been interesting to me. You know, I think it's like 85% of mammalian species retain the ability to make amyloid. Even zebrafish make amyloid, like these ancient animals, which seems really strange that all these animals would retain the ability to just make this, pathological plaque with no function that, causes disease. It makes a lot more sense if a lot of species retain the ability to make it because it does something, right? And that just always seemed to make sense to me. And it also, if you look at the Alzheimer's space and you just look at these trials, where now I think there's been dozens, almost hundreds of trials where they've just been creating drugs that just remove amyloid from the brain, and that's it. And they failed. Like they fail. One kind of got through, but on the whole, these amyloid removing trials are failing. And so it can't be that simple that amyloid's just bad and removing it is good. There must be some reason it's there. And I just I think that it's infection. So
Yeah.
there's it's it's a there's a huge amount more research that we can do. One of the teams in our network even published recently showing cytomegalovirus in the gut and the vagus nerve, and the brain tissue of people with Alzheimer's disease. So again, showing that you might have like passage of pathogens from the body to the brain over time as people age, which makes sense. So yeah, I think it's a absolutely where the research in Alzheimer's should be. And I think once we finally accept this is happening, it will be a paradigm shift that will change Alzheimer's. Yeah.
So Dr. Prowl, give us some hope. I mean, what you're doing
Yeah.
is awesome. Tell us what you're currently working on and what your hope is for
Mhm.
the future of the Lyme and COVID community over the next 5 years.
Yes. So I am optimistic just because there's so much talent in this space and there's a huge amount of development. There's a lot of work, especially in the diagnostics front, that just hasn't happened and that can. But we're announcing soon a big, long COVID diagnostics program focused on SARS-CoV-2 persistence, and innovating diagnostics for persistent virus, but other inflammatory markers that'll be out of UCSF. And we're building this starting to build the same program in chronic Lyme. And these are just next generation diagnostics programs with the best teams in the world. And I think that those programs, if run well, are going to really increase the number of tests that we'll be able to run on people to know, like I said before, not just that they have infections, but even sometimes what those infections are doing, or if they're derived from the central nervous system. All these questions that we want answered, there are platforms that can make this happen if there's enough effort. And then I think that what becomes really great is that once you start to innovate these diagnostics, you can start to really pull people into more targeted clinical trials. And we can begin to use them to really recruit in patients. Let's say it's a chronic Lyme trial. We can better know who has Lyme in the nervous system and who has Bartonella, but like the co-infections. We can sort this out in a way that I think is going to make our treatment trials and our treatment knowledge even more refined. And then I think we're going to be in a lot better shape. And so I think, I'm optimistic that as these efforts move forward, it's going to be like we're going to enter a time where there is a lot of rapid movement.
Yeah, well, Dr. Prowl, I can't thank you enough for joining the Tick Bootcamp podcast. I can tell you on behalf of the entire chronic Lyme community, we feel better because of the work you're doing for the future of everything tick-borne illness, not just Lyme disease. We need people like you. And I mean, clearly you're brilliant, you're driven, you're motivated, you're passionate, and you have pure intentions, which is really critical because sometimes people, are in it for the money. Let's be real, but obviously you're not. So we can't thank you enough for doing all the work that you're doing and taking time out of your busy life to come join our Tick Bootcamp podcast and to share all this really good information and hope with our listeners.
Well, thanks so much for having me. It was a really great conversation. Thanks for your interest.